To begin our examination of the nervous system tissue, we will look at the two anatomical subdivisions of the nervous system, which are featured in Figure 12.2 of the OpenStax textbook. The first is the central nervous system, which consists of the brain and the spinal cord. The brain is contained within the skull, and the spinal cord is surrounded by the bones of the vertebral column, specifically the bones that compose the cervical, thoracic, and a few of the lumbar bones, because in the lower regions of the lumbar and the sacral division of the vertebral column, there are only peripheral nervous system nerves that are located there. Which brings us to our second anatomical division of the nervous system, that is the peripheral nervous system, which consists of the nerves, which are essentially cord-like structures that are basically just collections of axons or bundles of axons that extend from sensory structures to the central nervous system, or are motor neurons that control muscles or glands that are extending from either the brain or the spinal cord to communicate with muscles or glands. We also have ganglia, which are not like bulges in nerves, typically where we see synapses between neurons and those nerves occurring. We also see sensory receptors in the peripheral nervous system. We'll have a whole entire module later on devoted to sensory receptors, so we'll examine those a little bit later on. This is Figure 12.2 from the OpenStax textbook, showing the two major divisions of the nervous system with the brain and the spinal cord on the upper right side of the diagram. And shown here for the peripheral nervous system, we see the ganglia, or singular ganglion here, as well as the various nerves that extend from the brain and spinal cord. Although in this diagram, they omit the nerves that are extending from the brain, but the brain does have several pairs of nerves that emerge from it, in addition to the spinal cord nerves that are shown here. We also see that there are functional divisions of the nervous system in which the central and peripheral nervous systems both contribute to these following three functions. And functionally, the nervous system will participate in sensory functions, or what is called sensory input, or just sensation for short. And sensory functions or sensation involves receiving information about the environment, so either the external environment or the internal environment. And the sensory functions of the nervous system are designed to detect changes or register the presence of changes from homeostasis. And these changes from homeostasis we call a stimulus. So really the sensory functions, the sensory input, are sensitive to stimuli, right? They're kind of paying attention to internal or external stimuli. And often what we see are that there are specific sensory neurons that are geared towards detecting certain stimuli, or sometimes there are specialized sensory receptors that are sensitive to particular stimuli. Often it's a combination of both sensory neurons and specialized sensory receptors that are detecting these stimuli. For the second function, we have what is called integration. And integration involves the processing of that sensory information, usually within the central nervous system. And integration is where the stimulus is compared to other current stimuli that are occurring, or that stimulus is compared with a past experience. And it's this combination of the stimulus, the other stimuli that are simultaneously occurring, and the past experiences that the nervous system uses to decide what the appropriate response or what the appropriate output should be. And so finally that leads us to our response, which is also called motor output. And the response or motor output is the body's response to that initial stimulus that was detected by the sensory neurons or the sensory receptors. And a response or a motor output usually involves the activation of muscles or glands, or sometimes both. So now that we've examined some basic components and functions of the nervous system, we can look more specifically at some of the cells that fulfill these functions in the nervous system, and primarily the cells that are fulfilling the three functions that we mentioned previously, the sensory input, the integration, and the motor output or the response, all three of those functions are usually fulfilled by neurons. And the neuron is featured in figure 12.8 in the OpenStax textbook. And the neurons are the communication cells of the nervous system. So they will create what are called electrochemical signals because there's an electrical component in addition to a chemical component. And those electrochemical signals are referred to as action potentials. So action potentials that are created in a neuron will then lead to the release of a chemical messenger called a neurotransmitter from that neuron. And it is the neurotransmitters that we're going to see stimulate and signal other neurons or other structures that the nervous system is communicating with. Examining some of the basic neuron parts, we see that the neuron has a cell body, or what is also called a neuron soma. And the neuron soma is the place in the neuron where the typical cell components are found. The neuron soma houses the nucleus of the neuron as well as most of the major organelles that we see in really any typical cell within the body. We see the mitochondria in the neuron soma, we see the endoplasmic reticulum, the Golgi apparatus, the ribosomes, and things like that. Now, from that neuron soma, we often have hundreds or even thousands of short branch-like extensions that are referred to as the dendrites. And the dendrites typically function to receive stimuli or information from other neurons or even other cells because they often will form chemical connections, chemical synapses with other neurons. So what we typically see is that dendrites have structures that are sensitive to stimuli, or those dendrites may contain receptors for neurotransmitters. So they will respond to those stimuli, or they will respond to those neurotransmitters that are released from other neurons. So, and as I mentioned, typically there's many dendrites per cell. Often there are a few hundred dendrites in a typical neuron, but there are certain neurons in the brain that have thousands of dendrites and are quite extensively branched. We also have a region that's kind of a cone-shaped narrowing region of the neuron soma that's referred to as the axon hillock. And the axon hillock leads to a single long process that's called the axon. And it's the axon hillock that is responsible for creating the electrochemical signal, the action potential of the neuron. So let's briefly examine those three parts there. So in this diagram, figure 12.8 from the OpenStax textbook, we see that this is the neuron soma here, or cell body, with the nucleus in the center there in purple. We see that there are a few dendrites extending from this particular neuron, but as I mentioned, there could be hundreds or thousands of dendrites, depending on where the neuron is and just the kind of connections that it makes within the brain or within other parts of the nervous system. And then we see this kind of narrow cone-shaped region right here, which is not labeled as such, but that's the axon hillock, and it's the axon hillock that leads to the axon. So let's examine the axon next. So the axon is a single and often very long process that extends from the axon hillock to little structures at its end called the axon terminals. And we see that even though there's only one axon in a given neuron, that axon may branch at its end many different times and have many different axon terminals. And what we'll end up seeing is that when an action potential is created along that axon, it will spread to every single one of those axon terminals that it has. We also see along the length of the axon, or we often see along the length of the axon, specialized glial cells, which form a structure called myelin. And myelin is a cell that wraps the axon at regular intervals, and it functions to permit or allow action potentials to travel down axons at a much faster pace, and allows them to travel much further before the action potential has to be regenerated. Because keep in mind that the action potential is a electrochemical signal, and it does have to be regenerated along the length of the axon. So if it is able to travel further before it has to be regenerated, then that allows action potentials to travel much faster down the length of axons, particularly if that axon is really, really long. And often myelin is formed by specialized nervous system cells called glial cells. In the central nervous system, we see that myelin is created by a glial cell called an oligodendrocyte. In the peripheral nervous system, myelin is created by cells called Schwann cells. Even though they have different names, they both fulfill the same function, just in the different divisions of the nervous system. We see that loss of myelin can often lead to conditions like MS or multiple sclerosis. MS is an autoimmune condition where the immune system kills the oligodendrocytes or the Schwann cells that form myelin along the length of the axon. And this can often lead to neuromuscular problems because it can create muscle weakness or loss of muscle control over most or all of the body. It's a very debilitating condition. We also see that adjacent myelin cells or adjacent myelin structures are not continuous with one another. There's often very small gaps between them, and those little gaps are called the nodes of Ranvier that exist between adjacent myelin wrappings on the axon. And what the nodes of Ranvier allow are they permit the regeneration of the action potential electrical signal along the length of the axon. And once we get a little bit deeper into the physiology of that, we'll see exactly how that works. Finally, at the end of the axon, we see the axon terminals, which are these little bulb-like expanded ends of the axon that function to release neurotransmitter, because the neurotransmitters are what are going to signal and communicate other cells or other neurons that particular neuron has synapses with. So let's examine the remainder of those structures in the diagram. So referring back to figure 12.8, we see the axon, which is often a very long structure, much longer than any individual dendrite. Along its length here, we see oligodendrocytes, so that tells us that this is a neuron that belongs to the central nervous system, because if this cell was in the peripheral nervous system, we would see that Schwann cells make up its myelin. We see the kind of regular wrappings of the oligodendrocyte pieces along the length of the axon, which form the myelin sheath. We also see the gaps between them, called the nodes of Ranvier. And then at the very ends, we see those little bulb-like structures that form the axon terminals, which form the synapses between this neuron and other neurons that it is adjacent to. So now that we've examined the neuron, we can start to examine some of the glial cells, or neuroglia of the nervous system. And there are 6 basic types of glial cells. There's 4 that are found specifically in the central nervous system and 2 in the peripheral nervous system. And they do have some overlapping functions with one another. Glial cells or neuroglia do not send signals or release neurotransmitters like neurons do. They don't create action potentials like neurons do, but rather we're going to see that they have other supporting roles in the nervous system. So from the OpenStax textbook, this shows just a handful of the glial cells or neuroglia that are located in the central nervous system. So we'll examine each of these cells that's shown here and what their specific functions are. So we'll begin with the glial cells of the central nervous system, starting with a cell type called an astrocyte. And the astrocytes are star-shaped cells, as their name implies, because the term astro means like star. So this is a star-shaped cell that has a number of extensions that emerge from their cell bodies. And these cellular extensions interact with nearby neurons as well as nearby blood vessels. So astrocytes function to remove excess neurotransmitters or certain ions like potassium in the space that's surrounding the neurons. So they essentially function to clean up a lot of the chemical messengers and other chemicals in the environment of the central nervous system. The astrocytes also function in maintaining what's called the blood-brain barrier. So they essentially stimulate the capillaries of the brains to essentially become impermeable to materials moving across them. And those capillaries prevent diffusion of many substances from the blood in those capillaries prevents them from crossing the capillary wall and entering the brain tissue. The blood-brain barrier does permit or allow the passage of things like glucose, which is the primary fuel for neurons in the brain. We see that it also allows passage of amino acids, oxygen, as well as the diffusion of oxygen and carbon dioxide across the walls of those capillaries. But pretty much every other substance and even other cells like leukocytes are prevented from entering the brain tissue by crossing through that capillary wall. So this is a protective function because the nervous system is very sensitive to changes in the chemistry of the environment of the neurons in the brain tissue. And so the blood-brain barrier really helps protect the brain chemistry from really any kind of even minor changes that are happening in the blood. We don't see those changes being reflected in the brain chemistry. So it keeps the environment of the central nervous system really, really consistent, even if blood chemistry or things like that are fluctuating quite extensively. We don't really see a lot of change happening in the chemistry of the central nervous system. So it's a very protective function. And it also makes it really difficult for cells that are in the bloodstream, pathogens and things like that, to enter the brain tissue and cause infections. But kind of the downside to that is that it restricts leukocytes from entering the brain tissue. So if there does happen to be an infection in the brain, like meningitis or something like that, that makes it notoriously difficult for the body to deal with. We also see cells called oligodendrocytes in the central nervous system. We've talked about this just a few moments ago. This is a cell that has a few branches or a few branching structures that emerge from it. And those branches tend to wrap around the axons of nearby neurons, and the oligodendrocyte wrappings form the myelin of the cells in the central nervous system, or the neurons in the central nervous system. We also see microglial cells, which are small glial cells that function to remove damaged or dead nervous system cells or tissues. And these also happen to function as leukocytes as well, because they fight infections within the central nervous system. And then finally, we see ependymal cells, which are cells that have cilia on their surface that function to move a liquid called cerebrospinal fluid through the brain's kind of various cavities and spaces. So returning to that diagram here, we see the astrocytes here with their many kind of little arm-like extensions that are kind of reaching out between capillaries and neurons here. We see a microglial cell here. We've already seen the oligodendrocytes, which wrap the axons at regular intervals to form myelin. And then these over here are the ependymal cells. So now we'll wrap up this particular discussion by looking at the glial cells of the peripheral nervous system. There's two types, two varieties, satellite cells and Schwann cells, and both of these have very overlapping functions with cells that we've already seen in the central nervous system. So the satellite cells are cells that surround the neuron somas in the peripheral nervous system. They function very similar to astrocytes in that they help to kind of maintain the chemistry of the environment of the peripheral nervous system, except that the satellite cells don't have the function of establishing a blood barrier, because we did see that the astrocytes had a function of establishing the blood-brain barrier in the central nervous system. Satellite cells do not establish a blood barrier between the capillaries and the structures that are present in the peripheral nervous system. And then finally, we see Schwann cells, which function a lot like oligodendrocytes in that they wrap axons of nearby neurons in the peripheral nervous system, and they form the myelin coating or the myelin sheath in neurons of the peripheral nervous system. The one main difference here with Schwann cells is that the entire Schwann cell wraps an axon rather than just part of that cell like the oligodendrocyte does. So examining those in the diagrams here, we see satellite cells, which are surrounding the neuron soma of this neuron that's located in the peripheral nervous system. And then we see individual Schwann cells that are wrapping the axon at regular intervals and forming that myelin sheath.